This is the principles of the innate and adaptive immunity. This is chapter 1.1 on myeloid lineage, and actually is the first lecture of a larger series of immunobiology topics, so please make sure to subscribe and follow along. This is Epitube Immunobiology, and let's begin. Hematopoiesis is essentially the process that hematopoietic stem cells will differentiate into our different parts of the immune system. This is the adaptive and innate immune system. Don't be afraid if you don't understand what's going on here right now. You will soon. Now let's begin with immunity. It really is to protect an individual from disease in 4 different steps. The first is immunological recognition. The second is immunoeffector responses. This is essentially to contain or eliminate the infection. The third is immune regulations. This is to make sure that we don't have too little or too much of an immune response. And lastly is immunological memory. Okay, so we need to remember previous pathogen infections because if we get infected with it again, we can mount a proper immune response to it quickly so that we don't have to get sick again. This essentially is the basis of vaccines. There's different lines of protection. 3 in particular. The first is the physical barriers. So this is the skin. And the second is the chemical. So they got antimicrobial proteins that are secreted on mucosal linings. This is like our gut linings, also our nasal passages, and also our skin. And last one, and by far the most comprehensive one, is the complement system. We'll mostly be spending our time here, as this would be the immunobiology set of all of this. You see here is actually the phagocytic white blood cells, or by text we're referring to as WBCs. That will recognize and kill microbes by releasing toxic chemicals and powerful degradative enzyme. So we have here is a neutrophil that chases the bacterium and actually phagocytizes it by eating it, okay? And then it actually releases toxic chemicals within the cell. And let's watch that one more time. All right, so white blood cells. White blood cells exist in both innate and adaptive immunity. Okay? All white blood cells are derived from hematopoietic stem cells, HSCs. This is the parent cell line, and they exist in the bone marrow. Okay? HSCs become the core components of the blood. They make essentially all the components of the blood. You see here the blood. They make red blood cells. Sorry, the HSCs make red blood cells. This is essentially to keep our bodies oxygenated. We got platelets to help clotting if there's any cuts in our body. And of course, white blood cells of both lymphoid and myeloid lineages. What we have here shown here is a hematopoietic stem cell that will proliferate and differentiate into different types of cell lines of both myeloid and lymphoid lineages. And these are essentially called committed cells when they can't turn back anymore and turn into other types of cell lines. Okay. So let's begin with myeloid progenitors. This lecture will be on myeloid progenitors, and the next lecture will be on lymphoid progenitors. But we have here the common myeloid progenitors are the precursors of the 2 different types of cells here, the immune-related ones and the non-innate immune-related ones. Innate immune system-related ones would be, you see here macrophages. We got granulocytes as well. And granulocytes actually have 3 different cell. We have 3 different cell types here. Okay, and we'll get to what these are in just a minute. And actually, that was referring to the chart earlier we saw. We also have mast cells, and of course, dendritic cells. In the non-innate immune-related ones, we have megakaryocytes. This would make, for example, the platelets and so on. It's a fancy name. We're just going to call these platelets. Okay, platelets. Okay, and you also got red blood cells. Okay. So let's get back to this chart here. I'm just going to fill it out slowly so that it actually helps us understand. Let's start with the hematopoietic stem cells here. Hematopoietic stem cells in the bone marrow, so I put the location of where they reside underneath in italicized font. So the pluripotent hematopoietic stem cells in the bone marrow, still in the bone marrow, can generate lymphoid progenitors and also myeloid progenitors. Okay. So lymphoid progenitors, we're just going to quickly browse through this because we won't be focusing on it. You'll see here you got plasma cells, activated T cells, natural killer cells. And so this essentially is going to be what we're focusing on next lecture. In this lecture, in terms of myeloid progenitors, we're going to understand the red side of the chart, This red side is all the myeloid progenitor related cells. You see here that you got the immature dendritic blood cells that can be made from the common myeloid progenitor. Okay. Also, this common myeloid progenitor can make the granulocytes and macrophages. These are actually progenitor cells. And also, they can also make megakaryocytes and erythrocytes. This is another fancy name for red blood cells. Of course, these ones then continue to differentiate into megakaryocytes and erythroblasts. Okay, I need to mention right here that I actually did include this terminology, this blast terminology. You'll see in some textbooks or websites, it essentially means that this is the precursor of the original, the final cell. So I'll show you here what I mean. The erythroblast eventually becomes an erythrocyte. Another name with this would be erythrocyte progenitor. Okay, so you see here megakaryocyte or megakaryoblast, you can say. This would be platelets. This megakaryocytes then make platelets for blood. Now these are the non-innate immune system related cells and components. That's what I was talking about in the previous slide. But on this side right here, we've got the granulocytes and the macrophages that can make these three types of cell lines: neutrophils, eosinophils, this one here, eosinophils and basophils. And then also that makes the unknown precursors of mast cells and monocytes. This is interesting. It's unknown, not that we know yet, or not that we're very sure, but it's still an unknown precursor of mast cells, and also the monocytes. Monocytes then can differentiate into macrophages. The mast cell precursors then make a mast cell. Okay, and then from the immature dendritic cells, what we have here is that these are in the blood from the bone marrow. Okay, they come from the bone marrow, go to the blood, and then they actually go to different tissues, and they're all immature and naive. Essentially naive meaning they don't particularly, they haven't recognized any pathogen or infected pathogen yet. But eventually, once they have, they become effector lymphocytes, essentially mature dendritic cells. Okay, so they go from the tissue to lymph nodes. Sorry. What we have here is that we're going to focus on these cell types. We've got macrophages, mast cells, neutrophils, eosinophils, and basophils, and of course the mature dendritic cells, because these components is what's the myeloid lineage in particular we want to focus on, and what is going to make our innate immune responses. Okay, so this is macrophages. Let's begin here. Macrophages are the precursor monocytes. My apologies. The precursor is a monocyte. They're really located in nearly all tissues. Monocytes continuously actually circulate the blood and enter tissues where they will differentiate into macrophages. Okay, and how they do this is particularly the function of which and how they perform is that they actually engulf and kill invading pathogens, as I showed you earlier, just like the neutrophil was doing so by chasing and phagocytizing the bacterium. The macrophage actually does the same thing. In addition to that, they also need to be very, very critical in orchestrating immune response by inducing inflammation, which is critical to mount a successful immune response. Okay. Essentially, this actually touches on point 3, where they secrete signaling proteins that activate other immune systems cells, perform antigen presentation, and then recruit them into an immune response. So essentially what this is saying is that they release certain chemicals and proteins, molecules that essentially recruit more proteins into the area of infection, and then so that it can help battle the infection. In addition, they also do what we call as antigen presentation. Okay, this is really important. Antigen presentation actually refers to them as antigen presentation cells. So this is when they actually eat the bacteriums. They express pieces of it on the outside. Okay, and this is antigen presentation. And so when they do so, they need to actually So let's begin here with macrophages. The precursor of macrophages is the monocytes. They're really located in all tissues because the monocytes continuously circulate the blood, and then they enter tissues where they differentiate into macrophages. Okay, and their function in particular is they need to engulf, or we could say phagocytize, and kill invading pathogens Like I showed you earlier with the neutrophil, they actually phagocytize the bacterium and digest it. They also orchestrate immune responses by inducing inflammation. This is critical for mounting a successful immune response because you want to make sure to recruit other cell lines, other lymphocytes to the area, and essentially by doing so, they have to. How they do that is by secreting signaling proteins. This tells other lymphocytes that there's an infection going on in this area to come to this area. At the same time, it activates the other immune system cells. And lastly, they also, very importantly, they perform antigen presentation. Antigen presentation we'll talk about a little bit more later, but essentially once it digests these bacteriums, and then it actually expresses some of them on the outside here, okay, of the cell, and presents, let me just say, it looks a little like Y shape. For example, a receptor that holds onto a piece of the bacterium right here, and then presents them for the the adaptive immune system downstream, and we'll talk about that later. And lastly, just a simple cleanup. They actually scavenge dead cells and cellular debris in the body. So they just go around and pick up, let's say, dirt and dust of the body. The other type of cell line here is the granulocytes. Actually, the granulocytes is an umbrella term for 3 different types of cell lines, cell types. Neutrophils, eosinophils, and basophils. So we have here the precursors for granulocytes would be the granuloblasts. Again, that terminology comes back. The granuloblasts actually has the granuloblast terminology here. So granulocyte precursor. Okay, they're located in the blood and the tissues. And particularly the functions of these, so I've actually had the 3 different types of cell lines here. You got the neutrophils. Firstly, the neutrophils have these granules in them as well because they have the cytotoxic chemicals and degradative enzyme in there as well. They perform phagocytosis of foreign pathogens, including viruses and bacteria. Eosinophils and basophils look quite similar. Okay, the nuclear shapes actually look a little bit different when you look at them under a microscope. But they perform similar functions as neutrophils, but particularly and primarily they're critical for killing of antibody-coated parasites. Okay, and although this is not completely confirmed, it is typically thought right now that it's too large for neutrophils and macrophages. So therefore, that's what eosinophils and basophils do. They actually attack parasites that have antibodies antibodies coated along the edge of the parasite, so that they can kill those cell lines because it's actually larger size. Note, relative to macrophages, granulocytes are actually very short-lived. They live for maybe 3 or 4 days, whereas granulocytes can live for weeks, and are actually produced in increased numbers during immune responses. So when an infection occurs, these neutrophils, eosinophils, and basophils, or essentially granulocytes in general, they actually are increased in higher numbers so that you can fight the infection. The other ones here is the mast cells. Well, the precursor of them I mentioned earlier, they're the unknown precursors. It's still not really well understood, so if anyone's looking for a fantastic paper to publish, or perhaps maybe even a Nobel Prize, go ahead. Look for this, the mast cells precursors. The locations of them, they're primarily at the internal surfaces of the body. So you see here, they are protected against parasitic worm infections. In terms of the function, what we have is that they release granules containing histamines and active agents, best known for allergic responses. As you can see, allergic responses often are related to what we have here is the histamines. Okay. And they often act as a first line of attack upon initial entry of a foreign pathogen. So if you got your skin, okay, and this is the outside, the pathogens right here, they can't get through the skin, but let's say they do, the mast cell may be sitting just right underneath to then attack the pathogen. So they're the first line, and they often are coating the internal surfaces of the body. And of course, we have dendritic cells. This is one of the most interesting and most complicated types of cell lines out there for the innate and adaptive immunity. The precursors are the myeloid progenitors, so we call them myeloblasts. What we have here is that the myeloblasts, they exist in the blood, lymph nodes, and tissues. Okay, so the dendritic cells actually will leave the bone marrow, mature in the blood, not even mature, sorry, they're inactive and naive, and then they go to the tissues, still naive, and eventually when they encounter a pathogen, then they become activated and they go to the lymph nodes. This will be in more detail later, but let's talk about the function. They circulate the blood and enter various tissues, okay, to ingest particulate matter by phagocytosis. Okay, so as mentioned earlier, they go to different tissues, eat up by phagocytosis different pathogens, and my apologies, and they continually ingest large amounts of extracellular fluids via pinocytosis. This essentially is like phagocytosis, rather it just kind of pinches off. So let's just say, for example, if you see here, this would be a massive one. But let's just say if this part right here actually pinches off with the other end here, then you actually end up forming an intracellular vesicle that came from the outside. This pinching would be pinocytosis. All right. The most important function here for dendritic cells is actually the antigen presentation. Okay, I mentioned briefly about that in macrophages earlier, but dendritic cells primarily are known for antigen presentation because after phagocytizing the product, the pathogens, they actually will present pieces of it on the cell surface to the T lymphocytes in the lymph nodes, and I'll talk about what T lymphocytes are later. And they are referred to, the dendritic cells therefore are referred to as antigen presenting cells. This is very critical downstream. And lastly, also very important, is there's a linkage between the innate and adaptive immunity. And the reason why I say that is important because T cells are most particularly known for their adaptive immunity. Okay, T lymphocytes are particularly known for adaptive immunity, whereas there is one type of T cell that's innate immunity. But dendritic cells are really only known for antigen presentation and, of course, making a link between innate and adaptive immunity. Okay. So dendritic cells form the difference between them. You'll see here that I said innate immunity. These neutrophils, granulocytes, eosinophils, basophils, and monocytes. This is actually macrophages. This is an innate immunity, including the dendritic cells. But the dendritic cells are really the primary function of the dendritic cells is also connect innate adaptive immunity. My apologies. Because what they do is they present little pieces of the pathogen to the T cells and B cells, and they tell these adaptive immunity to then mature over the next approximately 7 days to make your adaptive immunity. This would form your antibodies and, of course, different T cells specific for the pathogen. So in summary, this lecture talks about the physical and chemical barriers that serve as initial barrier of defense against foreign pathogens. Okay, the innate immunity is the first line of defense upon pathogen infection, upon passing physical barriers. The hematopoietic stem cells actually differentiate into myeloid progenitors. So we got here the neutrophils, eosinophils, and basophils, and dendritic cells. And actually, I must say that there should be monocytes and macrophages here. Monocytes, okay. And also the lymphoid progenitor cells that will then become mostly your adaptive immunity. And we'll talk about NK cells later. The granulocytes, which essentially is an umbrella term for these right here, neutrophils, eosinophils, and basophils, they phagocytose and destroy pathogens using these toxic cytoplasmic granules. So that's why you saw earlier in my animations that these three types of cell lines had a lot of granules in them. Macrophages and dendritic cells do the same, but they also digest foreign moieties for antigen presentations. These are to present the antigens to the the adaptive immunity on the cell surface. And dendritic cells are primarily responsible for antigen presentation so that they can activate the adaptive immunity downstream. Okay. So that'll do it for this lecture. This next lecture, chapter 1.2, will be on the lymphoid lineage, and particularly the innate immunity. So see how I give you a brief preview. Lymphocytes to the different types of T helper cells. So please remember to subscribe and to like this if you like it. You can also subscribe by clicking the Immunobiology EpiTube logo on the top. and we'll see you next time.